50 participants here, but yeah, anyway, let's just start. So, okay, good afternoon, everyone. Thank you for joining the Power Talk today. So my name is Gina. I'm one of the faculty member in biotechnology department of i3L. So thank you for joining. Today we are very honored to have Dr. David Booth. He's a senior lecturer from School of Life Science, University of Dundee. So today, his expertise, he's an expert in evolutionary biology. So today he's gonna share about introduction to statistical tests, which is, I think it's very important, especially for us. It's actually important in every field, but especially us who's working in life sciences. You do lots of experiment in the lab, but then after you get data, what to do next with the data, right? You need to interpret it with statistics. So today's talk will be very useful for you later, especially for your final project. Okay, so okay. So yeah, the presentation will be more or less an hour. So we will finish at 4 and will be followed by 30 minutes of Q&A. And at the end, to get the certificate, you need to fill the evaluation form. Okay, and yes, not for you, for the participants, but right, I'm not sure for the. I think for the speaker also get a certificate, right? But you
get a certificate.
I think so, but you don't need to fill the survey, the evaluation form. This is just for the attendees. Yeah. Okay. All so before we start, let me remind you again the code of the conduct. So the attendees are prohibited to activate audio, because we don't want to distract the other participants with the background noise during the Power Talk session. And the attendees are allowed to use chat feature to ask questions. So just please type your question in the question box. And then the evaluation survey at the end will be distributed to the Power Talk attendees at the end of the event. Yeah, so to get the certificate. Move on. Yeah, so just a little bit of introduction. So thank you, Dr. David Booth, for coming today. So just a bit biography, maybe you can add it later. So he graduated a BSc in genetics from the Queen's University of Belfast, and he finished his PhD in 2003 in population and evolutionary genetics at the same university, and he became a research fellow in 2003 at the Department of Agriculture and Rural Development, and in 2005 he's also becoming a research fellow at the Queen's University of Belfast. And interestingly, he's also a science presenter since 2008, I think, from at the BBC Northern Ireland. And he also a member of the Society for Conservation Biology, and also a Sentinel Science Ambassador for Northern Ireland. Next. Okay, shall we start? I would like to invite you, Dr. Booth. Please, the time is yours.
Yeah, no problem. I can just be full screen, can't I? Like
yes.
So it can all, and everyone can see me. Okay, everything's clear. Nothing's. It looks fine, I hope.
Yep. Yeah.
Yeah. Okay. That's excellent. Yeah. Thanks for the introduction. I've had a pretty non-standard career in science, and it's been fun. So I've worked for 20 something odd years, and I don't think I've had a single day where it felt like I was actually at work. It always feels like it's playtime rather than work time, which don't tell anybody, because they'll stop paying me my salary if they realize I actually enjoy what I do. So at the university, I teach predominantly genetics and statistics because that's kind of the core of, well, genetics is the base of pretty much every discipline and domain in biology. And statistics is it's the language that we use to speak to one another whenever we conduct experimental science. And not many people pay enough attention to it whenever they're, so it's one of these things. It's with all the skills that we teach in, well, skills and knowledge that we teach at university, there's a certain amount of, with the knowledge aspect of it, you're kind of filling your mind with facts and theories and theses and ideas, and that's kind of essential. But in order to push a field forward, that requires a whole different skill set. So you need to know the lay of the land, that's the knowledge, but you need to know how to navigate through the land and how to navigate into unknown territory. And in order to do that, you actually need to know a fair bit about how to design experiments, and more importantly, how to conduct the analytics that go with it afterwards to say whether you've actually seen a thing in the first place. And we have a module in level 2 called Statistics and Experimental Design, which I teach. And normally, from what I understand in other universities, students hate it. They hate this sort of aspect of things because it's not, interesting facts about cells and mice and great white sharks and bumblebees and things like that, or nerves and organs and the things that people will be interested in. So normally students hate this, but at our university, that module actually is one of the better liked in that semester. It doesn't seem to matter what they put up against it. I always win. So either it's the topic's interesting or it's a personality thing, like it's a cult. So I'm either training cultists or they actually enjoy what we do. Either way, it's absolutely essential. I mean, we've got chat window here, so we can use chat, can't we, to interact with one another? Like Catherine and Javita have both said something. Who else have we got on at the moment? Arlie, Novin, Natasha. Does everybody use chat, or is it just limited to the occasional person?
I think everybody can use chat. Are you comfortable with them interrupting you during the
of course,
lecture, or you prefer them to just write down?
No, they can.
Okay.
We can.
All right, so feel free to interrupt them during the lecture if you have any questions.
Yeah. We actually have, we've got a couple of different types of teaching activity that we do in the statistics modules. So we have the very standard, quite boring, well, I'll say boring, it's not that boring, sort of lecture where I address or I speak to a big crowd of students. But we like to keep the atmosphere fairly, not informal, but fairly friendly, so that if someone is following along the material that's being taught and they feel like they got lost, or they heard something and they didn't understand it, or they heard something and they understood it, but they didn't realize, the ramifications of it, they can stick the hand in the air and actually chat. We can have a discussion about it or chat with it, because more often than not, what happens is if one person's sitting there and they're feeling a little bit confused about what's going on, more often than not, the whole class feels a little bit confused about a topic. And there's three types of not knowing, or three types of confusion that we get with these sort of topics, because they are fairly complex in places. There's a confusion that you get because I maybe said something strange, or my accent, which is not, I don't have a normal UK accent, I don't think, but people didn't hear I didn't say it right, or I stumbled over some words, or, there's that sort of confusion. Then there's a second sort of confusion, which is you heard it and you understood it, but it's contradicted things that you've, understood before. So you're confused because you have your prior knowledge and then you have this new knowledge and they don't fit, they don't click together. And then there's a third type of confusion, which is the confusion that happens with statistics and experimental design, which is nobody knows the answer. You're the only person that's discovered an effect of some type, and you don't know how it works. So it's best if we actually do talk to one another whenever we feel confused about something, rather than politely waiting until the end and then being confused for an hour or 2 hours, and then at the end hoping that you remember all the things that kind of tripped you up throughout. So it's good if we can chat with one another. I think that's the one good thing of the pandemic, believe it or not, is the fact that we have this little chat window now. So whenever we're having sessions, I encourage the students to kind of put questions in and interact with one another as the session's going, so that we can all kind of move along together. So it's there are some positives and negatives to what's happened in the last year. So some sessions are like that, and then other sessions are entirely just interacting with one another. So there's no taught material. It's got some activities to do, and it's nice if you can. It's always handy if you can just pull me to one side and say, let's have a more meaningful chat about what we did this week. So yeah, if you have any questions at all, by all means use the chat, but I'm going to ask you questions as well. So what do we know about statistics at this point? Just to give a feel, what kind of statistics have we done? Like what's our baseline set of knowledge for statistics? What kind of things have we done in that domain or that area? Have we, I assume most people have so there's two aspects to it. And you can tell me whether does that look okay on the screen? Is that I
yes.
know it's just a red line that I've drawn.
Yeah, I can see the red line clearly.
Yeah. So on the one hand, we have what people think statistics are, which is a mixture of graphs and tables. And in the tables, we put normally means and standard deviations. So have we at this point calculated means and standard deviations? Is that something that we've done in mathematics or statistics for our biology? Just curious. I'm asking, obviously the students, of course, and you. Do you teach biology, or is it?
I teach lots of courses other than calculation related courses.
Yeah.
But yeah, we have lots of students here. Anyone? I'm pretty sure you have done this, right? Dafin.
Yes, we've done this.
Okay.
You've done this
several times.
Have you calculated standard error?
I've forgotten how to do it.
You forgot? No, you're not going to get quizzed on how to do these things. Don't worry about that. Or a thing called a confidence interval. Have you calculated that? So normally when we get students, that's what they think statistics is at this point. It's some form of a graph. So that's like a scatter, that'd be like an XY scatter plot, and that would be a best fit line or a line of best fit, or perhaps they've maybe even produced something like a bar graph with some form of error associated with it, and the little errors are expressed as whiskers. And more often than not, that would be the control, and that would be perhaps the treatment for the experiment. This would be our X variable, the thing that we've experimentally modulated. And this would be our Y variable, that's the thing that we've measured. So we think X has an effect on Y, and the little straight line corresponds to our model for how X affects Y. And we have an equivalent thing with the one over here, wherein we actually do have a little best fit line, and the little best fit line probably looks a little bit like this. And that's the effect that we think having a treatment on whatever it is that we're actually doing experimentally. The little blue line corresponds to the effect that we think that the treatment has relative to the control. So it's all very visual up until the point where you hit university. But that's really just a visualization thing more than anything. And it's more of a communication thing. So for example, when we conduct an experiment, we need to push forward our results to the wider world, the academic world. And we do that by preparing manuscripts. And those manuscripts need to contain all the key pieces of information that are required to understand what it is that we did. When we got the money to do the experiment, we spent it on the following things. This is our experimental procedure. This is how rigorous we think it was. And these are the results that we got out the other side. just in terms of raw observations more than anything. And it's more just the language of how we communicate with one another. But there's a whole other side to this, which is hypothesis testing. And I'm going to ask you a question. What have we done about hypothesis testing at this point? Have we tested any hypotheses, or have we conducted any hypothesis tests? Have we done any little experiments in the lab where we have set up a control and set up a treatment and check to see whether that treatment has had an effect? Is that something that we do or have done? No? You have? And what was it on?
I'm sure especially the fourth-year student, they should
I don't doubt it.
Started. Anyone?
You will have done some kind of little experimental manipulation of some type. So the kind of ones that we get students to do or pupils to do in the UK would be we take brine shrimp, or we take Daphnia, and because you can visualize, you can use a microscope to look through the effect of ingredients on food. Yeah, that's a pretty common one. With Daphnia, they're little freshwater invertebrates, and they're completely transparent. And when you put them under a microscope, you can see through them into their, you can actually see their physiology, working away. You can see the little heart pumping. And so you can expose the animal to treatments to see whether it has an effect on its circulatory system. So that's a pretty common one that we do over here. But yeah, ingredients in food's a good one, actually. We use that in a social science experiment in a different school, which is about palatability of food and how things interact with one another. So for example, we can set up a treatment regime, which is chocolate. Everybody likes chocolate, I assume. And we'll set up some kind of other fish. Do we like fish? Is fish a palatable thing?
Supposed to.
Yeah.
We live in archipelago.
Yeah. And then you can have something that goes with that. So that's just like two basic foodstuffs. So you could have, with that, you could have ice cream. And then with fish, I don't know, what would you have with fish? You might have rice, perhaps. That would be a reasonable thing to go with fish, right? And so you could set up a basic experiment where you have 4 treatment regimes, A, B, C, and D. So in treatment regime A, we have chocolate and ice cream. In treatment regime B, we have chocolate and rice, which sounds revolting, just even thinking about it. Although maybe it's not. Is there such a thing as chocolate rice? There must be. But we also have fish and ice cream, which sounds really revolting. So that's treatment regime C. And fish and rice, which sounds pretty reasonable in treatment regime D. And we could experimentally measure whether or not people like those combinations. And what we would find if we did that is that there would be an interaction effect occurring. So the people like chocolate, people like fish, people like ice cream, and people like rice, but whenever you put them into combination, you find that they're not quite so palatable when you start combining them together. So yeah, that's a kind of a common sixth form experiment that people conduct because it produces really nice results out the other side. Now, what do we know about hypothesis testing? Like, what do we know about the chain of testing a hypothesis? Have we done any experiments that involve collecting data to test a hypothesis, or do we know anything about the scientific method on that front? Let's make it a little bit more basic. What about the scientific method? What do we know about the scientific method?
Shall I randomly pick student then?
Yeah, if you like. I mean, if everyone's happy, I mean, is everybody happy to chat? You can just do it in the chat window. You don't need to speak out loud if you don't want to.
I think they're too shy. Come on. Come on, if you want to answer, please put your answer in the chat box.
This is believe it or not, this is the process of investigating something scientifically. Yeah, that's true. This is exactly the way all students are in the first week of university. It's really quite funny. But everybody gets over it fairly quickly because, you know, obviously we have like activities where we get to know one another and not be shy around one another. So yeah, it's the process that we use to investigate things scientifically. That's very true. And it's a chain of events that have to take place. So the first one is we have some kind of interesting observation about the world. So we see something about the world and it confuses us. It's we're not sure what's going on or what's causing it. So we have something that ticks a box to say this is something new. We're not sure what's going on here. We should probably investigate this a little bit more thoroughly. So, for example, an observation that was made in the UK about 40 years ago was that there was potentially a link between, so this is a biosecurity one, and there's a disease called tuberculosis. And tuberculosis is a respiratory disease that you get in mammals. Humans get it, but you also get it in cattle as well. So not only is it important for the health of people, it's important for the health of animals that we keep as livestock. And one of the animals that we keep as livestock are cows. We use it for milk and meat and things like that. And it was noticed that the cows were getting tuberculosis. It's called bovine TB. And people weren't sure exactly where the bovine TB was coming from, because whenever you've got an outbreak in a herd, you can cull the herd to clamp down on the disease, but then it would quickly reemerge in other places, which means that the disease must be coming from some other location. It must have some other host or some other intermediate host, or there must be some reservoir somewhere in nature that's causing the cattle to become infected, right? And pretty early on, people were seeing that the cattle that were being raised in the fields were actually interacting with wild animals. So shock horror, if you keep your livestock outside in nature, they will interact with natural things. And just to give you an example of what kind of natural things they can interact with, if we have a little cow, and I'm going to try to draw a little cow here. I don't know if it's going to look good though. Probably when I put the tail on. No, that looks like a horse, doesn't it? That does look like a horse. It now looks like a cow. There we go. There's our cow. It'll interact with multiple species. So for example, one species that it'll interact with would be ants. So whenever it's out in the field, it's going to be grazing on grass, and it's going to eat ants by accident. And there's a really sneaky life history strategy that's used by liver flukes. So we have liver flukes, which are little flatworms, and they use the ants as an intermediate host. And they use the ants in order to get eaten by the cow. The cow's liver is where the ant wants to live, so it can reproduce, to then produce a spore to become little little liver flukes that can produce spore. They then get excreted, that then reinfect the ants, and the cycle continues. So there's a chain of infection occurring there with liver flukes using the intermediate host of the ant. And what's interesting about that is actually it's a type of, has anyone ever heard of a parasite mind controlling its host? Anyone at all? It's called parasitic mind control. It's probably one of the most creepy and interesting biological things. So what happens in this case, this is fascinating, is when the spore of the flukes are in the ants, if the cows are not in the field, the ants will do their normal ant behaviors. Exactly, Cordyceps, yeah, Cordyceps Destructor, which is a fungus. Yeah, that's a fascinating piece of parasitic mind control. In this case, it's even weirder because when the cows are not in the field, the ants will behave normally. When the cows come into the field, the ants get mind controlled by the parasite to climb up to the top of flowers. Flowers. Yeah, that's one word that I actually have difficulty. Whenever the Northern Irish accent, we kind of butcher any word that is OWER at the end, and it sounds like I'm just saying the word R and nothing else. But anyway, when the cows are in the field, the ants climb up to the top of the flowers, and they bite down in the petals. And when they do that, the cows come across and they graze on the top of the little flowers. They eat the ants. The ants then release the spore into the gut of the cow, and that then completes the chain of infection. So that's a relatively small interaction that was occurring that was causing liver flukes to actually promulgate in the populations. But we get back to bovine TB. So these cows are interacting with lots of things. They're interacting with foxes, and they're interacting with birds and small mammals like mice and voles. But more importantly, they're also interacting with an animal called a badger. Has anyone heard of a badger?
I met once when I did my PhD in UK.
They're beautiful animals, aren't they? They're aggressive, but they're beautiful.
Yeah.
So they belong to the mustelid group. I can bring one up just so you can see it. They're very cute. They're gorgeous animals, actually. Let's see if we can find a picture of a badger. Yeah, that'll do. These are some gorgeous badger pictures I'm now looking at, and I'm going to show them to you. Let's see if this works. Badger pictures. Okay, can we see those okay? Yeah, okay, good. So these are they're a type of mustelid. You get them all over the world. They tend to form burrows. They tend to form little families. So they form burrows called sets, and they tend to have little families, and they are quite resilient creatures, and they can harbor infections. And what they found is that badgers actually are potential reservoirs for tuberculosis, because when you test the badger, you find that it carries TB, and it's a similar type of TB as the one that you find in cattle. And there was this observation that you had these two animals that both harbored the disease. Then that became that first initial kind of question: is there a link between these two? Does one cause the other? And there were a few pictures of cows and badgers interacting that kind of made the news. Especially this specific one here. Let's see if I can find it. There it is right there. Which sort of spurred people on to think that potentially what was happening is that the badger was acting as a reservoir, and it was either directly interacting with the cow, or it was interacting with the habitat of the cow, like, for example, urinating out the spore of the fungus, it's bacterium. And that was causing the infectious link. So a weird observation about the world then becomes an experimental, an idea for an experiment of some type to then test whether there is a cause and effect relationship between the two things. And that's the really important part, is that it's a causal relationship that one wants to know. So, for example, in this case here, a simple form of experiment that we could do in order to test this would be to design a simple control versus treatment regime, where we have cattle raised in a strong biosecurity environment. Let's say a part of the UK or part of the world where you don't find badgers, and we could have cattle raised in an environment where badgers are allowed to interact with them. Just to see whether there'd be TB arising as a measured outcome. in the actual the herds. So that's actually quite a nice example. It's not the best example in the world, but it's a nice one. So we have this idea. After we have our little observation, we now want to number one, propose some form of a hypothesis. And in this case, the hypothesis is that there's a causal link, and that we think, let's get rid of that, my little iPad. We think that there's some kind of link that the animals are getting infected off of one another. And we design an experiment that in some way tests that hypothetical relationship that we think exists. And that experiment must be of the type looking for cause and effect. Have we heard that phrase before, cause and effect? We have. What have we done on the cause and effect front in class? Have we done any experiments on cause and effect? No, yes, no, maybe.
Come on, guys, in the chat box.
So, in a chat box?
Nobody wants to say anything. I think they're too shy.
Yeah.
Or they have no clue. Don't know.
So a definite cause and effect there is the presence of a chat box means people forget everything that they did in the last couple of years. Yeah. Or if I ask someone a question, it's like an instant mind wipe. That's fine too. So we have this proposal or this idea of an experiment, and the critical thing behind the experiment is we need to have some measured outcome, some response that we think is going to actually happen, something that we can objectively put a ruler to, and that ruler will tell us whether or not a thing has occurred. So if we apply heat to a system, we use a thermometer to measure the heat that's been put into the system. If we shine a laser beam on a thing, we have some kind of measuring device that can measure the photons that kind of get emitted. We need some measurable outcome. So for example, in the case of our little cow experiment, we might think, what's a reasonable measure? A reasonable measure might be number of cows infected. So we might count the number of cows that carry the infection. And in doing this count, we can contrast the number of either the proportion or the number of cows infected in a herd. And our proposal might be that in the presence of badgers, the cows might have a higher level of infection than in the absence of badgers. Does that sound reasonable to everybody?
Yeah, that's quite reasonable.
Yeah. So that wouldn't be an unreasonable thing. And another measurable outcome we might take might be, for example, let's say we wanted to be a little bit less invasive with the cows. We might want to test perhaps a biomarker that we would use instead of actually, let's say, dissecting the animal out to see whether or not the lungs are infected. So we might come up, have we done something like polymerase chain reaction? Or do we know anything about genetics or genetic testing? Have we done anything like that? We've done a little bit?
Yes.
So we might putatively say, well, instead of dissecting the cow, we could take a little bit of aspirate from the cow's lungs, and we could use polymerase chain reaction to see if we can amplify genetic markers for the tuberculosis bacterium. Does that sound reasonable as well? I mean, believe it or not, the world is kind of at the moment using polymerase chain reaction based testing techniques for the most part to see whether or not people are carrying the COVID infection, and they're not amplifying anything about the human's genome. They're hoping to amplify specific genes that are found within the genome of the virus, and that would be a different type of measured outcome. That might be some kind of test. PCR test of some type, and the measured outcome in that case might be, for example, say the concentration of the product, so the concentration of the amplified product, or it might be the presence or absence of a band, a band on a gel, which indicates that something has been amplified. So there are lots of potential measured outcomes that can occur in all of these cases. And probably the trickiest part of coming up with an experiment is actually thinking about a good form of measured outcome, one that actually directly relates to your initial hypothesis. And believe it or not, it actually takes 3 or 4 years to really start to get good at this sort of thing. And to get really, really good, you probably have to do like a master's or a PhD after the fact. But it can be quite tricky. So that's our experimental aspect to it. We then revisit our hypothesis after the fact. So we then decide, do we or don't we reject or accept our initial hypothesis? So this is the final part of the chain. We've conducted our little experiment and we've determined whether or not something's actually occurring. We have some kind of measured outcome and we have some kind of idea of what the effect might be. And we're checking to see whether or not we see that effect actually occurring. And if we see the effect, we're going to accept our hypothesis. And if we don't see the effect, we're going to reject our hypothesis. And there's a taxi outside. I hope you didn't hear that. Did you hear that?
Yes, but that's okay.
Yeah, unfortunately, the world that we live in now, I would love it if I was actually teaching from one of the big lecture theaters. Like, I love those environments. They're like, it's like my hunting ground. At the moment, I'm teaching from my living room. Let's see if I can.
So do you work from home every day, or you go to the office like every so and so?
No, I work. So literally sitting in my lounge at the moment. Although I'm going to show you a thing. We have time, don't we? I mean, we're about halfway through what we're doing. So
okay.
I got this really, one of my ex-students who's now working down in Imperial College on bacterial, he's doing, it's something to do with brewing or baking. It's like a bacterial biotechnology thing. So he's looking at all these strains of bacteria. He sent me a picture on Facebook like about a week ago, and he showed me this like absolutely gorgeous little like sort of ornament. And he said that it would be perfect for me because I teach evolutionary biology. And this arrived in the post yesterday in the middle of a session. So I was teaching a session, and then I got this these like 3 little monkeys with and they're kind of sitting on they're sitting on Origin of Species, Darwin's Origin of Species.
Oh.
So it's nice,
that's
isn't it?
Funny, yeah. But why are they like closing their eyes?
Well, that's because one sees no evil, one hears no evil, and one speaks no evil. It's a UK thing. Maybe it's a world thing. I don't know. I actually just liked it because it's monkeys and chimpanzees and Darwin. But yeah, so this is how we teach now. So
we can talk about it further in another occasion. Probably it's very interesting. I like Darwin.
Of course.
Yeah. Okay.
We could do an entire session just on evolution if you like. That's
going to be very interesting.
Do you know what? Whenever I was your age, I came from Northern Ireland, and it's a very, very religious, like Christian religious part of the world. And a lot of the people that live there don't have a way of integrating science with their religious outlook, and they reject modern science, lots of it, in fact. So they don't think vaccines work, and they don't like modern medicine, and they don't like evolutionary biology and genetics, and basically anything that's been discovered in the last 200 or 300 years, they reject because it's modern. And I was always fascinated and passionate about the natural world. And whenever I saw documentaries by David Attenborough or Richard Dawkins, it just made me more interested in how living systems actually function. And that sent me off on a path where evolutionary biology was the only thing that I wanted to know more about. And I went to university thinking that was the only thing I wanted to actually do. And I didn't realize there was all this other stuff associated with it, that it's not just about learning about facts in books, that you actually have to, that a science degree is about, it's a modern apprenticeship and like discovery, like how to discover things. and that there's all this other interesting stuff that you learn the facts, but we're also going to teach you this skill set that allows you to look at the world and discover a new thing, like the process for actually discovering a new thing. And that's why I've ended up teaching a lot of statistics, because that's like it was a combination of the two. But yeah, I'd be more than happy to do a session on evolution. I'd like to say I love evolution. That's this 20, 30 years now, and every day I just learn something new and interesting. But we're getting off topic, like seriously off topic. So we come back to our
hello,
our little hello. Hi, Dina.
I think she forgot to switch off.
Hi, sorry. Yeah, I just forgot to mute.
We actually had a session towards the end of last semester where a student forgot to turn their mic off completely, and I think they might have went out for a walk because they just left the mic running, and you could hear people in the background the whole way throughout. So we would mute the microphone, and every now and then we would check in to see if they were still there, and they weren't, but the people were still there. It was very funny. Okay, we're getting off topic. So we're on this idea of rejecting and accepting hypotheses again. How do we? So let's say we've seen an effect. Is there a chance that that effect could have just been a weird observation? Let's set up a basic idea here. Let's say we have proportion of cows, or percentage. Let's do proportion actually from 0 to 1. Let's say we have the proportion of cows infected. This is going to be our control plots, and these are going to be our treatment plots. And in our control plots, we have this number of cows infected. So this is just a little bar graph. And in the treatment plot, we have this number infected. And when we look at these two things, we would definitely say there's a difference. Would you agree with that? There's definitely a difference between the control and the treatment here. You can say yes or no in chat. I think there's a difference, or there isn't. Either one's fine. And let's say we only looked at one field. So we're only going to look at one field for the control and one field for the treatment. What do you mean by depends, Catherine? What are you thinking?
It depends on how many samples and what is the standard variation.
Very good, very good, very good. So a critical thing that we need to know here is how many observations we've actually made. So in this case, I've only got one on the left and the right, but as I increase the number of unique observations I make of both the control and the treatment regime, I increase my understanding of what that population of observations might look like. So what do I mean by that? Let's say we increase it to 3 observations in each. Certainly I'm going to get a mean value, whatever that mean might be, or I'm going to get a centrally tendent value of some description. But I now can express not only what the centrally tendent value is, but how much spread in that value actually exists. So now I draw in the uncertainty or the error associated with the observations. And now what we see is a very different story. So initially, when we only saw the left and the right, we said they were different. We'd only made a single observation. But now whenever we have multiple observations, we see actually, even though the means are a little bit different, the error actually overlaps by quite a large margin. Does everyone see this? Now, the way we have to interpret this is those lines, those are normally either your standard error or your confidence interval. They're both technically the same thing, more or less. They're a little bit different, but not dramatically so. When we see these lines, we have to say, well, the mean might be here, exactly there, but it can exist anywhere in this range. It could be anywhere within that point. I think it's at the center, but it could be anywhere within that range there. And we say the same thing about this side as well. The mean can exist anywhere within this range. And so what we see here is this overlapping section in the middle. This overlapping section that I'm about to draw in green. So we see a huge amount of overlap. And because there's a lot of overlap, we cannot say for certain that the means of these two things are different from one another. Does everyone agree with that? Like, for example, on this side, the mean could be here, and on this side, the mean could be here. And in actual fact, it's an inversion of what we observed. In fact, I would say in this case, Joan, they are not significantly different at all. They're identical. We have no way of distinguishing between the means of these two groups, even though when we plot the graph, they appear to be different. Is everyone happy with that statement? Now, let's say I change it up a little bit and I make my experiment a lot more rigorous. So now instead of looking at 3 things, I'm going to look at 30 things. And the effect of increasing the sample size is one of reducing the amount of error in my observation. And what would probably happen in this case is as I increase my sample size, those little errors actually become smaller. And now we have a scenario where our experiment is powerful enough to distinguish between the two things. And now we see that there isn't actually an overlap anymore between them. And now we would say that it's likely that these are statistically significantly different from one another. Would everyone agree with that? That the control group has a lower infection rate than the treatment group? Would people be happy to make that statement? Yeah? Good. Now, what's interesting here is that this visualization that I'm showing you right now, this idea that we can measure the centrally tendent part of a sample and express the error on that central tendency, and that we can contrast two things just by using these error bars. This is the basics of the first hypothesis test that we actually teach people, which is known as a t-test. Has anyone heard that term before? A t-test? Some people have. Perfect, good. So the t-test is, we teach it in the second year, like the mechanics of how the whole thing works. And the reason why we choose this is because it's the simplest of the bunch. And if you understand this coldly, like if it makes sense for you, if you get how the mathematics work a little bit, and if you have a reasonably good feel for how shifting the parameters a little bit can change the core output, all other statistical tests fall into place because you have this visualization right here, and you know the numbers within a t-test correspond to certain kind of pictures that you might get in terms of your visualization. Now, what do I mean by that? So this basic test can then be expanded into all statistical tests. So if you get this one, then it all falls into place. It's like you don't even need to know the specifics of a thing, because they all have the same basic kind of structure or same basic language from start to finish. So what do I mean by that? Well, with statistical tests, especially when we're doing some form of experiment, we have some kind of mathematical method, some sort of mathematical method of calculating or estimating this delta. So this is this would be our little delta here, the difference between those two things. And we would express that difference relative to the amount of uncertainty that we have. All right, so there's our little, and I'll draw the uncertainty in green. So we divide that by our uncertainty. And in doing so, what happens is we're going to take that delta and we're going to scale it. I have 10 more minutes. Doesn't time fly? Yeah, I can do this in 10 minutes. Or we just make it part 1 of a 10-part series.
Maybe we can discuss more during the Q&A session afterwards.
Yeah, of course. I can even finish early. That's not a problem too. So when we look at our little statistical test here, we have our delta, which is our difference between the two groups. And we have our uncertainty, which is in green right there. And if we divide one of these things by the other, we actually get a thing called a test statistic. Has anyone heard that term before? A test statistic. No, yes, no? Okay. In the case of a t-test, the test statistic is known as t. And you may have seen the actual equation that goes with this. So the equation for t is the mean of group 1 minus the mean of group 2, all divided by the standard deviation pooled. Have you seen that little equation or some variation of that little equation before? Some may not. I think so. But look at the structure, look at the basic structure of it. So this is how we calculate that value t. This is this statistical ruler. What we have is the difference, so the delta. We have the mean of group 1 subtracted from the mean of group 2. So that just tells you the difference between the bar graphs. And then underneath, we have the standard deviation pulled between the two. And that's an expression of the uncertainty. Now let's think about what happens to t. So if we hold that idea of this little equation over here in our mind, we're going to look at some basic scenarios. The first scenario we're going to look at is one where they are very similar to one another, like this. So there's group 1, and that's going to be group 2, our control and our treatment. And here, what we see is our little delta is actually quite a small number. So the difference between the two groups is small. And here is our uncertainty, and I'll draw it in green. Our uncertainty is actually quite large. So what happens when you divide a small number by a large number? What happens when we divide a small number by a large number?
It's approaching 0.
It's approaching 0. That's exactly right. So it effectively becomes 0 or very close to 0, right? We'll set up a second scenario. And in our second scenario, we're actually going to see a very big effect. And here we have quite a large delta. And here we have an incredibly small uncertainty level. And so now we have a bit of an inversion. So now this is going to be large and this is going to be small. And what happens when we divide a small number by a large number? Or when we divide a large number by a small number? Well, the number will be big. Let's just call it big, question mark. Everyone happy with that? No, yes, no? Good. So now we have a basic idea of a statistical ruler, and we can say, well, when we calculate t, if the value is very small, we don't think we've seen something. And if the value is big, we suspect that we have seen something, that there's probably something going on in there. And that's the core tenet behind all statistical tests. We set some kind of form or formal ruler, which allows us to measure whether or not an effect is occurring. Now, the real question that people have is, at what point does it flip from being we haven't seen something to being we have seen something? And this is known as our, well, we would call it our alpha. But in this case, we have a kind of a threshold value of t. Threshold value of t. And if our experiment goes past that threshold, we would say at that point, we have observed something statistically significant. Have we covered that? Have we looked at t-tables yet? No, yes, no? Some people have, some people haven't. Okay, so in the remaining 5 minutes, we'll talk about how it is that we actually, how it is that we set that threshold then. Now, the way we set the threshold is actually interesting because it relates to the philosophy of science. Has anyone heard of Karl Popper? Has anyone heard of the philosopher Karl Popper? No. So this is, has anyone ever gone down a rabbit hole whenever they're on the internet, where they look at something and then they're on Wikipedia and they look at another thing and then they realize after 2 hours of just trolling across the internet, they're now looking at Greek philosophy? Has anyone ever done that? Or they're on YouTube and all of a sudden they realize they're looking at cat pics, videos of cats doing things. So if you ever want to go down, I think it's don't tolerate the intolerant. Yeah, that's true. But Karl Popper's big contribution was actually about setting up the basic mechanics of how we can interpret the world. And the core one was a thing called falsification. And this is a real, this will break your brain. Scientists can never prove a thing, ever. We can only ever say what doesn't work, or we can only ever reject things, but we can never prove something conclusively. The way you can do, or the way you can think about it, is think about two hypotheses that sit in contradiction to one another. We'll have a positive hypothesis and a negative hypothesis. And the positive hypothesis is, let's say there's a tiger. There's a tiger at school. So that'll be hypothesis one. There's a tiger at school somewhere. All right, so we'll call that our HA. And the second one that we're going to set up is a thing called there's no tiger at school. And that'll be our HO. And what we have here are two hypotheses. One of these hypotheses can never be disproven, and one of these hypotheses can be disproven. And we can never have a hypothesis that sits and can never be rejected or obliterated or iterated on. And what do I mean by that? Well, let's imagine this scenario here, the tiger being at school. We could set up tiger traps, and we could look for that tiger until the heat death of the universe, where no tiger could potentially exist, never observe a tiger, and at the same time, never reject that hypothesis. Does everyone agree with that? Like, there's no way for me to say there's a different version of this. There's no way for me to eliminate. Whereas this one here, we can set up tiger traps. And when we observe one tiger, the first time we observe a tiger, we can reject that second one. Does everyone see how that functions? One of these can be manipulated or can be refuted. The other one cannot. And so this process of setting up a thing called a null hypothesis, which is what this is known as, a null hypothesis, is the core ideal for the point at which we accept or reject our test statistic. What do I mean by that? And we are coming down to the last minute, so we're almost nailed it on time, even though I've rambled a little bit in places. Remember, we have our little t statistic where 0 means nothing and a big number means something. Well, we set up a world called a null hypothesis, or a null hypothesis world. So we would build a picture of the t statistic, and we would say, well, if the t statistic is 0, we suspect that there's nothing going on. But we want to know what kind of values of t we could get in a world of a null hypothesis. And believe it or not, in a world of no effect, where nothing can happen, you actually do get quite a large range of t. More often than not, you get a value close to 0. So when we compare things and no effect can actually be occurring, we would get a t-score of 0. But very occasionally, because our experimental apparatus is not perfect, and because our samples might not be as random as we think we are, and because animals behave like animals and sometimes don't do what we want them to do, and 101 other reasons why there might be error in the system. Very occasionally, you might conduct an experiment and actually get quite a large t-score, or you might get a very small one, just purely by random chance, just purely because life isn't perfect. And so in this world of the null hypothesis, we use this to test whether or not our experiment has actually had an effect. And the way we do it is we say, well, in this world of the null hypothesis above my head, I will accept a t-score that is greater than or lesser than a threshold value. And we actually set up as a proportion. We say, if my p-value is less than 0.05, in this world of the null hypothesis, I can reject the idea of the null hypothesis and say that I've seen an effect. When I say 0.05, what I mean is I'm going to get rid of about 2.5% at this end and 2.5% at that end of my t-distribution. And if in my experiment, my t-score is greater than this or lesser than this, I've seen something interesting occurring. And when our experiments have been conducted reasonably rigorously, the t-score is approximately 2 at this end and approximately -2 at that end in terms of making an observation. And so that's the basic core of the idea. So if we go back to our little experiment with our little cows, wherever it was right here, if my t-score that I calculated here was greater than 2 or less than -2, I would be able to reject my null hypothesis and say that badgers actually cause TB. Spoiler alert, they don't. Just in case anyone goes away from the session thinking that they actually do. We've conducted many a trial over the last 30 years to check to see whether that actually does occur. And it doesn't. The little badgers are actually quite civilized on that front. Okay, I think that's bang on 10 o'clock at my end. Is that, I guess it's about 5 o'clock in where you are, right?
Yeah, it's 4 p.m. actually. Yeah.
Okay.
We are
was that reasonably interesting? That wasn't too boring, I hope?
No, that's quite interesting. Yeah, especially we usually during a normal webinar that we usually have, the speaker usually shows slides, but this is very interesting because you're using a Blackboard, and yeah, that's quite interesting.
If you prefer slides, we can do it that way.
No. I think that this is very interesting. That if people tend to get bored if you just show some slides, right? But I think this is quite interesting.
I mean, it's some people really. So whenever we have the actual sessions, what we do is we have the pre-reading material. We have pre-recorded videos. We also have PowerPoints, so like little skeletons of a lecture that a student can annotate as they run through the lecture. And some of my colleagues, they'll adhere very rigorously to the skeleton, and some of my colleagues will do that. All they'll do is just a Blackboard, and they'll interact with the class and kind of modulate the kind of lesson to the sort of the average of the class. It really depends. It depends on how big the class is and what kind of topic it is that you're actually teaching. But in some of the really higher level classes, so level 3 and 4 genetics, often we're just discussing research more than anything. So we'll maybe bring up a single figure from a paper that everybody's read, and we'll discuss that experiment. So yeah, it really depends. But yeah, if you had really wanted a PowerPoint, we could have done that.
No. This is very interesting and engaging, and especially because this is related to calculation. It's very easily to follow if you draw. Shall we continue to question and answer session? Okay.
Yeah, of course. Just ask questions. Yeah.
All right.
I
think anybody can just speak if you want, or if you're too shy, you can just type, because
I can actually show you a t-test if you want.
Okay. Yeah. While waiting for question to be typed here in the chat.
Yeah. I can just. So one of the things that we actually use at the university is a thing called R, which is a statistical programming language. So this is like the software interface for like if I ever want to plot graphs, or if I ever want to do any statistical test, we never do it on a calculator. I mean, I say we never do it on a calculator, but I actually have my my T83 plus right here that I frequently use. But more often than not, when we're doing work for like manuscripts or research projects, we have to use some kind of statistical software package because we're handling a lot of data. so no, I assume in mathematics you got taught how to use the statistical functions on a calculator, like how to store things in the memory. Did they do that with you? Like memory plus that thing? No?
I honestly never use it, but I don't know, maybe other participants here have used it.
Yeah, I mean, all of these little colors here, above they all correspond to a thing. You can actually do stats on it. But we would tend to use something like this instead. So that, this is our little calculator right here. So we teach all of our students how to do statistics in this fashion. And this is actually, so if you want to know how employable a person is, ask them whether or not they can do a little bit of stats using something called R or SPSS or S+, because those for like data science and scientists, it's like a super employable, employability goes up the more you engage with this sort of thing. And why did you divide the delta by the uncertainty? Why did it have to be those two values? Why do we divide the delta by the, okay, we can do that. There's two, so I'll get back to this in a second, but we'll go for this here. Any experiment we design has two sources of variation in it. The first one is called systematic variation. And the second one is called unsystematic. And I apologize for my terrible handwriting, but I've only had 1 cup of coffee today. It's very early. Systematic variation, that's the variation that we induce experimentally. That's the cause and effect part. That's I apply heat to the system, or I infect the cell line, or I expose the animals to that's our experimental design. The second thing is your unsystematic variation. And unsystematic variation is measurement error. It's well, because we're living things vary, right? So if I take any two random humans in the world and I compare them, they're different. We would agree with that, wouldn't we? Doesn't matter who they are or where they're from. If I take any two random people and I contrast them, they are not going to be the same. There's so much variation in the system, it's not even funny. And that applies to wild animals as well. And it applies to cell lines that grow in a laboratory. It applies to, like you could take a culture of bacteria, and you could grow the same culture, and you could grow it in 2 flasks with almost identical, so you've set up identical conditions in each, you're going to get a slightly different result, right? Like the bacterial culture one's actually a good one. So we're going to take the exact same amount of a culture. We're going to put them into the same flask type. We're going to apply the exact same amount of medium, and then we'll take a sample and measure the number of, we'll do like colony forming units. We use a microscope to look at the colony forming units. You're going to get a different value here than you are here because of unsystematic variation in the system. What do I mean by unsystematic variation? Well, it could be whenever we took the bacteria out and put them in, we actually took, instead of 10 microliters, we took 11 on this side, and we took 9 on this side, right? Even though we think we're taking 10, we're taking 11 on this side and 9 on this side by accident. So that's a source of unsystematic variation. My instrumentation has a certain amount of error in it. Regardless of how precise I think it might be, there's always a little bit of error. And so we are always designing experiments with these two things in mind. We want to design an experiment where we minimize this bit and we maximize this bit relative to one another. So if we have a really precise experiment, and let's say those are the differences between the means, a really precise experiment would look like this, where the differences are large and the noise or the error is small. And so all statistical tests are effectively telling you how much signal is coming through in your experiment relative to the amount of noise in your experiment, or how much systematic variation you have relative to the unsystematic. And so any test statistic is technically a measure of that. It tells you how noisy your experiment is and whether or not you can reasonably accept or reject a thing. So if we have a large amount of signal and a very small amount of noise, we get a small number. If we have a very small amount of signal and a very large amount of noise, we get a big number. And so this becomes effectively a measuring stick for how noisy your experiment is. Does that answer your question, Joan? Is that a reasonable answer? All statistical tests are like this, believe it or not. Like there isn't a single one that doesn't behave like this. So have you done a best fit line? Has anyone done like a line of best fit or a linear model? It's sometimes known as. So when we do a linear model, which is like X against Y, and they're both continuous. So you do a best fit line that looks like that. So this is your signal. But whenever we do it using R, we also get that second component, which is the noise. That best fit line could have been this, or it could have been this, or it could have been that. You know, there are a lot of best fit lines that are reasonable for the data that you might have. And so we actually have this component of noise as well. I don't know if you can see me drawing it in. And if, in the case of our little best fit line, the gradient, make sure I do this the right way around. If the gradient is greater than 0, when I add and subtract noise, we say that we've seen an effect here. So it's always a measure of signal to noise in experimental systems. I hope that answers your question. Okay.
Yes, that answers my question, sir. Thank you.
Have you done a chi-squared test?
I have.
A chi-squared, so we can do that here as well. A chi-squared is a signal to noise test too. So I'll do that. It's the observed minus the effect squared all over. So the observed minus the expected squared all over the expected, right? That's the basic idea of, that's how you calculate chi-squared. And if you're doing it, let's say, for example, with fly breeding or population genetics, you do the sum of. Right? That makes sense, that thing that I've just drawn, right? I mean, everyone's familiar with that basic equation, I hope. Maybe they might do it without capital letters, actually. It might look like this here, maybe. I don't know how it might look like that, perhaps. But let's look at what's happening here. So this is our observation, and this is our expectation. So the top part of this becomes our signal, the difference between what we observe and what we expect. And then the bottom part is effectively our noise. That's our expectations. There's some component down here to say how noisy the system is. So this again is a signal to noise ratio test of a type. We go on to learn other tests like F tests and things like that. And what you should see as you progress is they all have the same basic structure. They calculate a test statistic, and that test statistic is some way of saying how big an effect is that you've actually seen in your wee experiment. And once you get that locked down, once you realize that, you know, this is the kind of the core, it all kind of, things start to click, and it all, you know, then you can start, believe it or not, you can actually start teaching yourself this stuff, which is quite strange for me to see. So for example, the student that told me about the little ornament that I ordered, I actually chat with him quite a lot on Facebook now about the science that he does. And he's telling me things that he's learned or he's taught himself, which I'm stealing some of his ideas at this point because I actually find them quite interesting. So it's very strange to see the student becoming the master. But yeah, the goal is to understand mechanically how it works. And once you do that, you can actually start to teach yourself most of it, which is quite weird. Then I'm out of a job. Do we have any other questions?
Sir, may I ask further about the chi-square one?
Of course.
So you said earlier it's signal divided by noise, but why is it in chi-square? The noise is the expected value.
So in this case, it's a form of so the noise is a two-component part. The first thing is the amount of uncertainty, but that uncertainty is also the magnitude of the measurement. So in the case of, for example, I'll try to give you an example. that might work. Okay. So if I have my little chi-squared up here, right? So this is my observed minus expected. I'm not going to tell you anything about the expected just yet. All right. But I'm now going to draw in, or at least I hope I can draw in 2 examples that show you how it works in terms of noise. All right. So this is going to be, we're going to calculate chi-squared here. So on the first one, I'm going to say the difference between my observed and expected was 5. And I'm going to put a question mark underneath. Does that sound fair so far? Like we're not going to put anything into the expected underneath at the moment. You just see the difference between your observed and your expected. Okay, so just to see whether it's a noisy system or not. In experiment 1, my expectation was 6, and experiment 2, my expectation was 6,000. Do we see the difference between these two? So this is a relatively small experiment with not many observations, and this is a relatively big experiment with lots and lots of observations. So here we have a very noisy system, and here we have a system with a lot of precision associated with it. Does everyone see that, the difference between these two values? Another way to think about it might, so this is one where we actually see a difference between things. Let's say we have, this is my chi-squared for, so this gives me a chi-squared value of 0 or 0.5 even. Let's imagine we have an extremely large experiment, which gets that value of 0.5. It also produces an identical value of 0.5. But now we have a much higher level of precision associated with it. So the bottom part effectively tells us the signal to noise aspect of it. It's a small component of it is actually, so one of the things we do in second year is you have to actually go to class, or you have in class. One of the things that we get undergrads to do is actually design their own experiments to explore this idea. And it's related to a thing called statistical power. So it's one thing to be told how a thing works. It's quite another thing entirely to design an experiment to explore this concept. So whenever we have a little chi-squared, we get them to do small experiments. So n is 3 or 4. It's a very small number. We don't have many observations. We have relatively few independent observations, and we get them to design big experiments. And they more or less test the exact same hypothesis. And what they can see is that as you increase your sample size, because you increase your sample size, you're going to put more stuff. You put more effort into the collecting of the signal. You reduce the effect of the noise, and thus you see where you couldn't actually see an effect in the first instance because your experiment wasn't powerful enough. When you increase the sample size, when you change that ratio of the two, you actually end up in a position where you can make a positive statement of one type or another. I don't feel like I'm answering this very well. I don't feel like I'm answering this very well at all. What would be another way to think about it? What would be another good way to think about it? It's really one of these things where you have to do it to see how it works. You get told it, and then you do it a few times, and then it kind of clicks that that's how the statistical ruler actually functions. I apologize if that didn't sound very sensible.
I think I get what you mean. Yeah, I think it makes sense to me, at least for right now.
I mean, when you're doing it, think about it like this: when you're doing a chi-squared, all right, let's say we're doing a basic chi-squared, and we have, let's say we have 3 in one cell and 4 in another. All right, so this is a one-way chi-squared test. So that's n equals, like that's 7 things have been observed. We haven't observed many things in this case, but now you change it, you keep the ratios the same, and now make it like 3,000 over 4,000. So the number of independent observations you've made are much more dramatic, you still get the same chi-squared value, but the power of the observation becomes much more robust. We actually go through a set of formal calculations with it just to show how that works, how we can be very positive about one and not very positive about the other, because this would be a very noisy system, and this would be a very precise system as a byproduct. Yeah, it's interesting. So I take it you're designing some of your own experiments for your final year projects. Is that true?
Yeah, we have an ongoing project in statistics class.
And what's it about?
For me, my project right now is about blood groups. So actually, I'm also using chi-square, although it's not the usual kind of chi-square, the one with tables. It's actually the expected part is derived from Hardy-Weinberg principle.
Oh, right. Yeah. Okay. That's believe it or not, that's where I first learned chi-squared.
Same.
Yeah. So I did like my PhDs in population genetics, and Hardy-Weinberg principle is kind of believe it or not, that was the first piece of scientific theory I learned in first year at university, where I went, this makes perfect sense. I understand how the world functions now. It was like one of those light bulb moments. Yeah, so you use the chi-square to determine, in your case, whether or not the observed and expected counts of genotypes are in alignment with one another. Is that correct?
Yes, exactly like that.
Yeah, that's a really interesting example, actually. So you have your allele frequencies. Let's say you've got, is it a 2 allelic system like PQ, something like that, or big A, little a?
I'm doing 3 alleles because ABO blood group.
The ABO blood group, right? Okay, yeah, that's fine. So you've got some kind of setup for your blood group like that. You have your expected genotypes that are going to come out the other side. You can calculate the frequencies of these and determine whether or not based on the Hardy-Weinberg equation, whether or not the proportions that you're actually seeing. Where are you getting your sample from, actually? Your blood group sample? Is it like a public data set, or have you been given a data set?
We distribute a survey, basically asking students here what their blood groups are.
That's really interesting. So you've got so your observation is going to be, tell us your blood type.
Yeah, basically.
You're then going to calculate these, which are the frequencies of the alleles, and that's then going to feed into the expectation, and then you can contrast the two. That's really neat. That's quite a cool little experiment. I like that. Yeah, that's funky. What do the results look like? Have you gotten to the stage where you're analyzing the results yet?
Actually, I am analyzing the genotype proportions instead of the individual allele frequencies. I think we did not reject HO though.
It would be really surprising for me if your null hypothesis could be rejected, because think about what has to happen with Hardy-Weinberg for it to not be in equilibrium. You need to have assortative mating. So there would have to be some mechanism by which people with an A blood group could identify people with an A blood group and only mate with them. Or people with an A blood group could identify people with a B blood group and go, ugh, do not want to mate. And I don't think that exists. I find it very, that would be a weird thing if you picked your future wife, husband, partner on the basis of their blood group or blood type. So that'd be one that would have to be in place. You have to come from a very small population. So that's another one of the principles, so if that assumption is broken, then things can go out of equilibrium. And most people live in fairly big populations. They come from, even if they're in little towns, those towns are part of bigger environments. Also natural selection. So some form of selective pressure would have to be occurring on blood groups. And it's my understanding that it doesn't matter whether you're an A or a B, especially not in modern life anyway. Both have an equal kind of fitness, so to speak. So it'd be shocking to me if your blood group was out of equilibrium. There are some markers that could be out of equilibrium, but I don't think blood group would be one of them. But that's good. That's good. So you all of the assumptions have probably been met. Interesting. I would love to see the results actually. To see what you actually got. That'd be kind of cool. We did a similar experiment at school, but it was using peas. So we looked at the genetics of peas that have been grown in a field, which was kind of quite good fun. We used a little alizyme marker, like a genetic marker, which was diploid to determine whether or not the population of peas was in equilibrium. That's kind of cool. I like it.
Thank you, sir. Maybe if you want to see once we have submitted our final report, I can email it to you or something.
Of course. I'd be happy to have a look. Always interested in these sort of things, especially cool little experiments like that. Yeah. Just get Putu to find some way to send an email, and I'll be more than happy to have a look at what you did.
I see. Also, your explanation makes chi-square make more sense to me.
Yeah, but think about with how chi-squared works. If your observations and expectations are the same, your difference is 0. So it doesn't matter how big the denominator is. You're always going to get a chi value of 0. And as your observations and expectations get more different from one another, then your chi value gets bigger and bigger and bigger. And obviously it hits a threshold, at which point you say, I've got to reject this null hypothesis. The chi distribution is a little bit different than the t distribution. It goes from 0 to infinity. And it kind of looks a little bit like this, a little bit. So it's, and you've got that same idea that above a certain threshold value of chi, when your p-value is less than 0.05, so this proportion of the distribution remaining, that's the part at which when we've seen that much of an effect, at that point we say we have to reject our null hypothesis. Normally it's a chi-score of about 4 or more for most experiments. What chi-score did you get? What chi value did you get?
I don't remember, but it was small.
It wasn't 4. It was small, right?
Yeah.
Yeah. That's cool. I'm impressed. I like it.
Thank you, sir.
Oh, no, thank you. Any questions about what it's like to be at a university, or what the weather's like in Scotland, or anything else? I don't want to be boring you, Sol. I would hate to be boring you.
Maybe those who are planning to go to UK or Scotland to continue master, or maybe do PhD, you can ask questions.
Of course.
We have 1 more minute though.
We have 1 more. So it'll have to be a quick question. Yeah, a PhD in biology is it's a lot of fun. So my PhD was a mix of, so there was a lot of population genetics. My PhD was a mixture of 6 months of the year I was in a laboratory, and then 3 months of the year I was out in the field collecting biological samples. So like invasive algae, fish stocks were a big one that we were looking at. We also did a little bit of work with badgers, and so there was a lot of field work that went with it. It was really, really good fun. In fact, everything about university was quite good fun. I really enjoyed my undergraduate degree as well, because of the amount of stuff that you learn and the amount of skills that you get. And I have no regrets, absolutely no regrets. About half of my friends went into industry, and the other half went into academia. And it's always interesting to find out what my friends from university get up to.
Maybe the most frequently asked question by the students is whether they should go to academia or they should go to industry. What do you suggest them to do?
I mean, it's so my friend Marshall is he actually works for a company called Randox, which is in Northern Ireland, and they're a drug discovery company, and he loves it there. But it's a very different type of ethos. So they're very much you get paid a very handsome salary to do this sort of work in an industrial environment. Whereas I like freedom. So I like the academic freedom aspect of things. I want to explore ideas that I'm interested in. So in the last few years, I've moved slightly away from genetics and into social biology. And one of the things that we're interested in at the moment is a thing called nature connection. I don't know if you've, so I feel very connected to nature because of my upbringing, but some people feel completely disconnected from it. And so we're exploring the social biology aspect of it. You know, what are the causes and what are the manifestations of it? So if you want the freedom to look at, you know, to do different things and explore different things and explore ideas, go into academia. But if you like the idea of earning a lot of money, go into industry, I guess. It all depends. It depends on what you want.
Okay. All right. Any more questions, guys? If there's no more question, maybe you can just drop you an email probably. Yeah. So thank you very much, David, for sharing today. It's very insightful, and yeah, we really enjoy your presentation. I hope to see you in the future lecture, probably about evolution or more advanced statistic, probably.
That'd be kind of cool. We could just talk about plants and animals and cool things. That'd be fine. Or we could actually do more stats, either one, you know.
Yeah, we will be in touch with you.
Thank you so much for your time, everybody. I hope you have a great day.
Okay, have a nice day.
You have a nice day too.
Thank you. So for those who are waiting for certificate, don't forget to scan this barcode. So you will be directed to the evaluation form. Don't forget to fill it. And yeah, so I hope you're having a nice day. Yeah, thank you. Bye.